So with all this study of evolution and talk of a common ancestor, it really begs the question then, where or how did life originate, right? When we draw these phylogenetic trees, we talk about everything from bacteria to single-celled eukaryotes, plants, fungi, animals, but they all lead back to a common form of life. We all are made of cells. We all have DNA as our genetic material. We all have some fundamental biochemistry that is common to all life. All of that suggests, in the light of evolution, that we originated from a common population, right? Our three domains, our biggest categories of life, all lead back to some earliest organism. So where and how did that earliest organism arise? And the short answer is we don't know. Now, we can conjecture, but we'll never really know because we can't go back and look. So even if we were able to spontaneously create life in a test tube, which we haven't been able to do yet, I suspect someday we will be, it doesn't mean that that's how life originated on our planet. Right? It could be some other chemistry. Early chemistry of Earth was very different, right? And that's why we need to consider that life was very different when it first arose. Now, there are a series of experiments, very famous, the Miller-Urey experiments from 1950s, that basically showed you could take inorganic compounds and under conditions where they suspected were similar to early Earth, could create the building blocks of life, organic compounds. And these were great experiments. They were able to show that they could produce basically lots of different monosaccharides, lipids, nitrogenous bases, amino acids, from the kinds of very basic inorganic chemicals like ammonia and methane and hydrogen gas that they were thought to be more prevalent on early Earth, right? That's pretty amazing that all these things that we talked about earlier in the quarter as the building blocks for life could arise spontaneously under these conditions. Now, as we've continued to study this, it's probably more likely that it was conditions similar to what we find in these alkaline thermal vents at the bottom of the ocean. These are the conditions that we probably saw life arise about 4 billion years ago. Now, there's a great book, and the first chapter of this book, which is all about the origin of life, is posted on the website. But that book is called Life Ascending by Nick Lane. And he breaks down 10 great inventions of evolution that he's seen over the last 4 billion years and puts them in fairly accessible language. So this may seem a little thick, but it's a great chapter, and I encourage you to read it, though I won't hold you responsible for it. But one of the things that he talks about is that when we look at, and if you go on in biology and science, you'll study what's called the Krebs cycle, and this is one of the fundamental biochemical pathways by which we break down our food. He's like, if you look at the molecules that are arising spontaneously, these are molecules that are most likely to arise just thermodynamically. Right? From the laws of physics, these are the most likely to pop up. And these are the molecules that we see as intermediates in this pathway. And we know, again, based on thermodynamics, that gradients are important, that concentrations are important, that if we could you can drive this reaction forward now to break those things down to make energy for food, or if they're in abundance, you can drive them backwards to build up biomacromolecules. Right? So what we see here are lots of indications of early chemistry that is biochemistry. Now, there's another hypothesis that came along that's called the RNA world hypothesis. And this then brings in another variable of RNA, that we know we can spontaneously form RNA monomers from early Earth conditions, and those monomers can polymerize randomly. Now, the cool thing about RNA that we haven't talked about, not in any great detail, is the fact that it can act like an enzyme. So all of a sudden, you have a molecule that we call a ribozyme. So ribozyme is RNA enzyme, right? It's a ribozyme. And that ribozyme is then a molecule that can catalyze chemical reactions, but is also an information-containing molecule. So if one were to arise, which you can show this in a lab, that you can get this to happen in a jar, you can get an RNA molecule that catalyzes its own replication. And as soon as you have something replicating, and a process by which that replication can be tweaked, i.e. you get mutations to occur. If you then select for those molecules that replicate themselves better, boom, you've got something going here, right? Something that can be selected for. So how do you put all these pieces together? Well, that's where we have kind of the least information. So you could think about some way that RNA and polypeptides could interact to help catalyze each other. You have some enzymatic reactions, like kind of a biochemical soup, that if some of that can get captured into a lipid vesicle, which we know that bilayer, that vesicle, will form spontaneously, again, based on hydrophilic interactions, thermodynamics. If you could capture some of that biochemistry inside there, you have kind of a protocell now. Now, how that cell evolves to reproduce itself and how the chemistry aids in the survival of that vesicle, that's a little bit unclear, but it's not inconceivable. But that's about as good an explanation as we have, which, in all things considered, it's not that great an explanation. Now, that being said, we do know and have evidence, that points to life did probably evolve from a single population. And again, over those billions of years, lots of cool things have occurred. And what I want to stress here is that life's biochemistry is very much affected by Earth's conditions. But life itself is such a powerful force, it can affect the Earth as well, terraforming. So there's a couple of dates that I really want you to know, and it's just a list that we'll go over in class, but these are dates that you want to commit to memory, right? So you want to know that the Earth is 4.6 billion years old, right? That life first arose in prokaryotic cells, probably somewhere between 4 and 3.8 billion years ago. Now, when we talk about life transforming the planet, you can talk about photosynthesis. And about 2.7 billion years ago, we saw oxygen levels start to rise. And we know that oxygen is a byproduct of photosynthesis. And we see in the fossil record the appearance of what we call cyanobacteria, photosynthetic prokaryotes. So Earth is 4.6 billion years, life 4 to 3.8 billion years. 2.7 billion years ago, we see photosynthesis arising, and oxygen levels go up. And it took about 500 million years of photosynthesis by those prokaryotes to get oxygen levels where they are today. Now, that increase in oxygen affects the biochemistry of other organisms, and there's all kinds of great stuff you can study in that. But we're going to call single-celled eukaryotes around 1.2 billion years ago. And then the earliest multicellular organisms around 1 billion years ago. Colonization of land about 500 million years ago, and then our species, we're going to peg them at about 200,000 years. Okay, so those are the dates I want you to know. But you can see here, as we look at this graph, 3.8 billion years ago is the origin of prokaryotes. And then as we move, we see photosynthesis occurring, oxygen-producing photosynthesis about 2.7 billion years ago. We see 1.2 billion years ago true eukaryotic cells arising. First mitochondria and the endosymbiotic event, and then a second endosymbiotic event with chloroplasts, lineage of plants and animals, right? So again, plants really kind of moved onto land about 500 million years ago, and then again, our species, just a blink of an eye, 200,000 years. It's nothing, right? So commit those to memory, and we'll discuss them further.